Happy Darwin Week. Hooray. Thank you for coming. I never know what's going to happen on the first night of these things. People are already leaving. It's very depressing. It's going to be really good. Yeah, okay. Sure you do. That's what they all say. So what I'm going to do with my little 15-minute slot is talk about some animals in association with Darwin that most people don't think about. If you think about Charles Darwin, if you know anything about what he worked on, probably the first things that pop into mind are the things that Dan talked about: the Galapagos finches, the Galapagos tortoises, vertebrates. But Darwin was arguably one of the last of the really complete biologists. He wasn't even just a complete biologist. He was interested in geology and all sorts of things. But he worked on all sorts of stuff. Dan had a quote from Mistletoe. We probably can all have a quote from Darwin in our talks. Do we all have quotes from Darwin in our talks? Okay, most of, all right, some of us do, and the rest of us could if they wanted to. So I'm going to talk about Darwin's interest in invertebrates. And little known fact, Darwin's first ever published words in a scientific publication were on insects. And he's got this great quote from his autobiography that I had to put in here. But no pursuit at Cambridge was followed with nearly so much eagerness or gave me so much pleasure as collecting beetles. I will give a proof of my zeal. This guy was a real biologist. One day, on tearing off some old bark, I saw two rare beetles and seized one in each hand. Then I saw a third and new kind. This is the collector's dilemma. What do? You don't have enough vials. You don't have enough hands. What are you going to do? Which I could not bear to lose, so that I popped the one which I held in my right hand into my mouth. Alas! It ejected some intensely acrid fluid, which burnt my tongue, so that I was forced to spit the beetle out, which was lost, as well as the third one. So he really got screwed by this. So when I say his first ever published words, I had to sort of parse things the right way. Basically, what he did was he contributed to a volume on British insects, and he told the author effectively, I found this particular species of moth in Cambridge. And his comment about that in his autobiography was, no poet ever felt more delight at seeing his first poem published than I did at seeing in Stevens' Illustrations of British Insects the magic words captured by C. Darwin, Esquire. And anybody who's ever had a scientific publication, your first one, that's how you feel. And there's his. I found this species. Okay, it's a little bit off. It's supposed to be higher than that. It's the one right above it. It's that one. I don't know how it did that. Graphophora plecta, Cambridge, C. Darwin, Esquire. And I'm falling apart here. There we go. And this is the species. It's got a different genus name, Ochopleura plecta, which basically means yellow-shouldered. So you can see those sort of cream-colored, yellowish stripes there. This is a noctuid moth that he just told Stevens was in Cambridge. And those were his first published words in a scientific publication. He did more with invertebrates. Who knows what these are? What are these? Barnacles. These are barnacles. Darwin was a barnacle expert. He spent several years developing expertise in barnacles. He published an immense monograph on barnacles. He really wanted to become an expert in a group of organisms. He thought that was really important. And he thought, barnacles, there's some cool stuff going on there. It'll probably take me a year or two to work it all out. It took him 8 years. It was a nightmare. It was a huge mess. But his work is still cited today. Many of the species he described are still valid, including this one, Balanus improvisus, Darwin 1854. So barnacles, sessile crustaceans, sessile marine crustaceans that are little filter feeders that have all this strange morphology. He did not do this drawing. George Sowerby did it. I would have been I'm already impressed with Darwin, but if he had done these drawings, I would have been really blown away. All right, so getting a little closer to my own interests and areas of expertise. Great Perplexities. Here's a quote from one of Darwin's letters. This is actually his first letter to Alfred Russel Wallace. The theory of natural selection is properly known as the Darwin-Wallace theory of natural selection. They sort of came up with it independently and put their work together. And in his first letter to Wallace, he said, one of the subjects on which I have been experimentizing and which cost me much trouble is the means of distribution of all organic beings found on oceanic islands. And any facts on this subject would be most greatly received. Land mollusks are a great perplexity to me. And this is only one of many letters in which he mentioned the fact that Land mollusks were driving him mad. Land snails, terrestrial gastropods were driving him absolutely crazy. And I guess the next question is, why? Why did he care? Why does this whole question matter? And the reason it's important to him is he's very interested in how plants and animals and really all organisms got to be where they are today, because a major component of his theory was descent with modification. Living species arose from pre-existing species. They had an origin. So that means they had a geographic origin. They started somewhere. There was an ancestral mammal species. There was an ancestral bird species. There was an ancestral land snail species. Where was it? Well, it had to be somewhere. Well, then how did it get to all these other places? And if you're talking about birds or even large mammals and you're talking about continental things, it's no big deal. They can just move, right? Over millions of years, they could just disperse and diversify, no problem. You know what happens to snails and slugs when you pour salt on them. You probably did it as a kid if you were mean. You took a slug and you put some salt on it and they shrivel up and die. So Darwin thought, how do snails get out to archipelagos like the Galapagos? How do they get out to Hawaii? How do they cross thousands of miles of salt water? That should have killed them, right? So this is a big problem for him, because under his theory, they couldn't have just arisen there through spontaneous generation or something like that. They had to evolve from ancestors, and they had to get out to these islands somehow. So this is a big problem for him. He came up with two possible explanations. Can you think of what they might have been? How could snails get out to islands? Something carried them. That's an easy one, like birds, right? I've never seen a land snail quite that big, but you know, that's one possibility. And this happens. We know this happens. You can find land snails and even clams sometimes stuck to the feet of birds. And birds go all over the place. They can easily traverse some of these great distances. The experimentizing he mentioned in that last slide was based on his other idea of how land snails could have traversed these distances. They could have floated. Land snails are very good at basically hibernating. They can pull themselves into their shells. They can put a little coating on the opening of their shell. Some of them have an operculum, a little sort of shell door that they can use to close the opening of their shell. And maybe they could just float across these distances. So he would literally take land snails and put them in buckets and crates of seawater and leave them there for days or weeks and then come back and pull them out and see if they were alive. And quite often they were. So he said, gosh, you know, they can survive immersion in salt water. So Darwin was really perplexed by land snails, but he thought he had some explanations for how they got up to some of these islands. Does anybody know where this is? Not Fountain Bluff. Inspiration Point. This is LaRue Pine Hills. This is one of the most amazing areas in Illinois, and if you're a slow and careful driver like I am, it's about 45 minutes that way, because you kind of have to do that and go down 3, right, unless you go through the back roads. It's really close. And this is a fascinating place, and all of this whitish stuff here is limestone. It's made of calcium carbonate. Land snails love calcium carbonate because they use it to make their shells. This area right here is the highest diversity of land snails and highest abundance of land snails in all of Illinois, and quite possibly most of the Midwest, because there's lots of limestone there, and the snails love it. So we've been out there and have been doing some work on this species in particular. This is the carinate pill snail, Euchemotrema hubrichti, and I was going to try to find some shells in my lab and pass them around like Dan did his mistletoe, and I forgot. So afterwards, if you want to go see their shells, I can take you down there and show you. They're, as adults, they're about that big or so, which doesn't seem like it's very large, but it's pretty good size for land snails. And they've got these really cool sort of lens-shaped shells. Carinate refers to a carina, which is a keel. It's the sharp little edge. Some of them you could almost cut something with. That edge is really sharp. So here they are alive, and there's roughly where LaRue Pine Hills is, northern Union County. So if we zoom in on that bluff line, you can tell it's a really steep incline because here are all these topo lines just smashed together. That's a cliff, right? What we did was we went out to LaRue Pine Hills because the U.S. Forest Service wanted to know something about this land snail. They knew it was there. They didn't know anything about it. They couldn't tell us anything about where it was exactly, what it looked like. They'd never seen one alive. So we got some money from them to go out and find it and tell them where it was. So I went out there with a couple of students. And we climbed all over these bluffs. And actually, a couple of SIU undergraduates found the first sort of high abundance site for this species, the one area where we found lots of them, which I've just named C and D there. It's kind of in the middle here. We looked all up and down this bluff line. The entire range of this species is about 4 miles in length. It's LaRue Pine Hills and a couple of outcroppings just to the north of LaRue Pine Hills, as far as we know. It's never been found anywhere else. So this is what we call a highly restricted endemic. It's only found here at LaRue Pine Hills, nowhere else on Earth. This is it. If you want a candidate for state mollusk of Illinois, there it is, right? So let's look at some of these habitats. This is Site D, so this is one of these high abundance sites that a couple of my students found several years ago. This is what they really like. They like these limestone areas with all these sort of cracks and stuff back in here. There's nice moss and lichens up there. They probably feed on lichens, and they hide back in these cracks. During the summer, when it's really hot out there, they will estivate or hibernate, basically. They'll pull themselves into their shell and stick themselves to the underside of some of these big limestone slabs. If you pick one up, you might find 30 or 40 underneath there just hanging out, waiting for it to get nice and cool and damp again. So this is one of these high abundance sites. This is another one. I called these the stairs, but because I'm a scientist, I can't call them stairs. I have to give them like a letter name. So I just called it E. But I thought of it as the stairs because it was like a bunch of steps going up this bluff. We're sort of facing northwest here, and that's the pond, and the Mississippi River's out that way. And you can climb up there, and you can find the snails up under these little limestone slabs. So again, this entire range is from these two sampling sites. Well, A down to G is about just over 3 miles. The whole range is a little bit longer than that. The whole point of this is we also did some genetic work. So we sampled a bunch of snails from these different areas, and we looked at their mitochondrial DNA, and what we found was very strange. What we found is that this high abundance site that I just called F here, which I'll show you in a second, was genetically distinct from all the rest of them. I didn't really know why this would be the case at first, because it's kind of right in the middle. We sampled a couple individuals from down here, not very many. We didn't find many down here. But these guys were different from all the rest of these populations. They were genetically distinct. Not very different, but slightly different and consistently different. So why? Well, there's Site F. That's where population F is. Population F, you climb up sort of the back side of this thing, and you get up here, and they're up on the top of this little outcropping here. And there are maybe a couple down on the sides, but they like this stuff up on the top. That's their preferred habitat. So why is F, or why do I think F is different from everything else? Because to something that size, that's an island. They don't like, as far as we can tell, they don't like this other stuff. They don't like regular old dirt. They want to be up on the tops of these limestone outcroppings. Well, this little outcropping is separated from the main bluff line. Not by very much, but by a little bit. To us, we could walk the distance in a minute or two. That's a long distance to a snail this big. So the upshot of this is effectively an island of habitat. It's not an oceanic island, but it's the same sort of thing. I'd like to think that Darwin would think this was really cool. I don't know. But here you've got an island, no oceans involved, but it's an island of habitat. Is this something that we need to conserve or protect? Did I run off to the Forest Service and say, this is a different species or anything like that? No. It's just an interesting find that we don't have just nice gene flow sort of all along the length of this. There are little breaks within that, even that short little range. All right, so Euchemotrema huberti is a polygyrid land snail. It is a member of the family Polygyridi. Polygyridi has about 300 living species. They are all North American. It's what we call a North American endemic. So it's only found in North America. North America, of course, includes Mexico. Can't forget that. And there's lots of diversity here. Some of these guys get relatively big, maybe that big. Many of them are even smaller than the carinate pill snail is. And some of the species are hairy, like this one. This one you can find not too far from here. You find this in Tennessee. They have hairy shells. This one is from probably either California or Oregon. I think Oregon. Hairy shells. Why do they have hairy shells? No idea. They just do. So it's a very diverse group, and surely you can read all of these names, and you're going to take a lot home from this slide. The point of this is just to say, we're not just working on Euchemotrema hubrichti, we're looking at the whole family, and that's where Euchemotrema hubrichti fits into this. This is a molecular biology, just a molecular estimate of the tree of life, or the subsection of the tree of life for polygyrids. And there is our local, highly restricted endemic right there in green. All right, switching gears just for the last couple minutes here. Charles Darwin trivia time. Graduate students and professors cannot answer this question. What was the fastest selling book that Charles Darwin ever wrote? The one that flew off the shelves? The Origin of Species? No, not The Origin of Species. Any other guesses? Can you name any other Charles Darwin books? It's that one. The Formation of Vegetable Mold through the Action of Worms and Observations on Their Habits was the fastest selling book during his lifetime. There are a couple of caveats to this. First of all, it was published in 1881, which was shortly before Darwin's death. At this point, Darwin was already famous because of The Origin of Species and everything else he had done. So there were probably people that said, hey, Darwin's got a new book out. It's got to be great. Darwin wrote it. It's got to be amazing. So that probably helped a little bit. You know, he was kind of a celebrity by then. But he had been interested in and working on earthworms for decades and had wanted to put all this together into a book for a very long time, and finally was able to do it before he passed away. We do research on annelids here at SIU. We have a project called WormNet2, Assembling the Annelid Tree of Life. I'm a co-investigator on that project with lots of other people, both in the US and in Europe. And the whole point of the WormNet2 project, the whole goal is to use genetic data to figure out the entire tree of life for all living annelid worms, all segmented worms. So annelids are things like earthworms, leeches, and lots of marine bizarre things that you have probably never heard of, but you're going to see a couple of pictures of here in a minute. So there are anywhere between 18 to 20,000 described species of living annelid. And if you are a good, free-thinking, tax-paying citizen of the United States of America, there ought to be one question in your mind right now. Why? Why would, because this is your tax dollars at work here, folks. And I'm going to tell you, you're spending it on a lot stupider things than this. So don't be too angry. Why would anybody be interested in the annelid tree of life? I'm a phylogeneticist, which means what I do is reconstruct parts of the tree of life. I find the tree of life inherently interesting. I would do it just for that. But there are other things that you could do with this. We could ask questions like, how did the different annelid lifestyles evolve? There are lots of different ways in which these segmented worms make their living. Some of them are parasites. You all know what this is. It's a medicinal leech. We've got these marine forms up here at the top. I guess I should use this pointer. These are predatory. They feed on other small marine invertebrates. Maybe you've seen some of these. The feather dusters, filter feeders. They live in tubes, and they stick these little fans out, and they catch little particles of food. Detritivores, basically deposit feeders. This thing, this red thing, not an annelid. That's a sea urchin. But these white things underneath here, those are tentacles from a spaghetti worm. So what it's doing is it's sending out its tentacles, kind of like what that one's doing, but it's sort of feeling around on the bottom and finding lots of good gunk that floats down to the bottom and feeding on that instead of trying to grab it out of the water. All the way to things like these up here on the top, the vent worms, the vestimentiferans, that don't really feed at all. They have bacteria living inside their body that process chemicals coming out of these hot vents. So how did all these evolve? You can't even ask that question if you don't have an annelid tree of life worked out. You could make up stories, but to have a real leg to stand on, you need to know how these species are related to each other. The last question, it would not be a good biology talk if there wasn't a picture involving sexual reproduction of some sort. So here's your obligatory sex slide. This is two earthworms mating. They're hermaphrodites, so they're swapping sperm. So they're lined up and passing sperm to each other. And just to end with a quote from almost the last sentence of Darwin's book on worms, "When we behold a wide turf-covered expanse, we should remember that its smoothness, on which so much of its beauty depends is mainly due to all the inequalities having been slowly leveled by worms. It is a marvelous reflection that the whole of the superficial mold over any such expanse has passed and will again pass every few years through the bodies of worms. The plough is one of the most ancient and most valuable of man's inventions. But long before he existed, the land was in fact regularly plowed and still continues to be thus plowed by earthworms. It may be doubted, and this, when I read this as an invertebrate zoologist, I nearly weep with pride. It may be doubted whether there are many other animals which have played so important a part in the history of the world as have these lowly organized creatures. So I'll stop there, and I don't remember who's next. Thank you.